So, Assalamualaikum. My name is Nur Atika. So, today I would like to talk about hypersensitivity type 2. Okay, so the content that I will be presenting today is introduction, definition, stimulant that triggers of the hypersensitivity, and the fourth one is the mechanism and also the stimulants. So, the first one is the hypersensitivity reactions are immune responses that are exaggerated or inappropriate against an antigen or allergen. There are four types of hypersensitivity reactions categorized by the mechanism and time course of the immune response, which is the first one is the type 1 is immediate, type 2 is cytotoxic, type 3 immune complex, and the type 4 is delayed. So today we will focus on type 2 hypersensitivity, also known as cytotoxic hypersensitivity, which plays a critical role in various autoimmune and alloimmune diseases. So, definition of hypersensitivity type 2 is that it involves antibody mediated destructions or alteration of the cells. These reactions are typically mediated by IgG and IgM antibodies directed against antigens on the surface of target cells or extracellular matrix. So the immune system mistakenly targets normal cells, leading to their destructions or dysfunctions. So, what can triggers of the hypersensitivity reaction? So, the first one is the autoantigens, which is the antigens that are normally present on the body's own cell in autoimmune diseases. The immune system erroneously recognize these self-antigens as foreign. So, as an example, autoimmune hemolytic anemia, where the red blood cells are targeted by their own cell. So the alloantigens is the antigens from other individuals of the same species. So it is common in transfusion reactions and transplant rejections, such as, for example, is hemolytic disease of the newborn, where the fetal red blood cells are attacked by the maternal antibodies. And for the third one is exogenous antigens, which is the foreign antigens such as drugs or microbial proteins that bind to the cell surfaces, making them targets for the immune system. Such example is the drug-induced hemolytic anemia, where medications bind to red blood cells and it triggering an immune response. So the next one we go to the mechanism of hypersensitivity reactions, which is the first one is the antibody productions, which is the IgG and IgM antibodies bind to the specific antigens on the surface of target cells. These antigens can be intrinsic to the cell membrane or absorb exogenous substance. So the second one is complement activation, which is the antibody antigens complex activates the complement system. And the complement cascade results in the formation of the membrane attack complex, which creates the pores in the cell membranes leading to the cell lysis. The next step is the opsonization and the phagocytosis, which is the antibodies and the complement fragments opsonize the target cells. And the phagocytes, such as macrophages and the neutrophils, recognize these opsonins via their FC and complement receptors, leading to the phagocytosis and destructions of the opsonized cells. And then the next step of the mechanism is that the antibody-dependent cellular cytotoxicity, the NK cells will bind to the FC region of the antibodies, attach to the target cells through their FC receptors. So this binding will trigger the NK cells to release the cytotoxic molecules such as perforin and granzymes, which induce apoptosis in the target cells. Last but not least, for the mechanism is that interference with cell functions. In some cases, antibodies can bind to cell surface receptors, either blocking their normal function or mimicking the natural ligand, leading to the abnormal cells signaling. So examples includes the antibody stimulating the TSH receptor in graft disease or blocking acetylcholine receptors in myasthenia gravis. So this is the example in the graft disease and also myasthenia gravis. So in the conclusion, type 2 hypersensitivity or cytotoxic hypersensitivity is an antibody-mediated immune response targeting antigens on cell surface, leading to the cell destruction or dysfunction. These processes involve IgG and IgM antibodies and mechanisms like complement activation and phagocytosis. So triggers includes autoantigens, alloantigens, and exogenous antigens, contributing to conditions such as autoimmune hemolytic anemia and hemolytic disease of the newborn. So that's all for the hypersensitivity type 2. Thank you.